Everyone. My name is, I guess I'll use the doctor, Dr. Lisa Galea, and I'm the lead of the Women's Health Research Cluster. And I'm really delighted today to introduce you all to one of my favorite people, Dr. Deena Walker. And I know she's going to give us a fantastic talk on thyroid hormones. And I don't know very much about thyroid hormones, so I'm excited about that. But before we listen to her, I want to recognize that we live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Toronto, which is covered by the Toronto Purchase number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And I know not all of you are in on Turtle Island, so I encourage all of you to learn about the Indigenous peoples on lands of which you dwell. A little bit about the Women's Health Research Cluster. We have actually over 1,000 members now across 43 countries, which is pretty amazing, and we are here to promote and catalyze women's health research across all disciplines. We run a number of events, a trainee research presentation series, so if you're a trainee and interested in presenting, please look out for calls for that. We run a number of travel awards. We just had an award lottery. It's a lottery system, unfortunately, now, but hey, you never know. So they run twice a year. So please be on the lookout for that. Become a member and support us if you're not already a member. And with that, I'm going to stop sharing my screen so that I can read out Dr. Walker's bio. How's your computer doing? Is it in the middle of your presentation?
It's back. It's fine.
Yay! Okay. Um, so while you start sharing, I will read your bio and your fun fact.
Okay.
I made, listeners, I made Deena change her fun fact for you. Deena Walker is an assistant professor in the Department of Behavioral Neuroscience at the Oregon Health and Science University. She's passionate about understanding how hormones influence brain and behavior, so am I, and is dedicated to using her diverse training background to identify precision-based treatments for neuropsychiatric disorders, particularly for women. Currently, her lab is focused on how hormones influence sex-specific vulnerability to alcohol use disorder and substance use disorder. She moved to Portland, Oregon in 2020, an auspicious year, after doing her postdoctoral fellowship at the Icahn School of Medicine at Mount Sinai, where she studied the molecular mechanisms of sex differences and addiction. As a PhD student at the University of Texas in Austin, she studied how the brain controls ovulation and menopause with a specific focus on hormonal regulation of gene expression. Such exciting work. In recognition of her innovative and impactful science, she has been awarded several early career awards, including the Frank A. Beach Early Career Investigator Award from the Society for Behavioral Neuroendocrinology in 2023. In addition to her laboratory work, Dr. Walker is active in science outreach and storytelling. Her stories about the impact of her own life experiences on scientific questions that have been featured on the podcast StoryCollider. Now, her fun fact is that she was the lead Cinderella in a fourth grade play, or if you're in Canada, we'd say grade four. Is that correct? Was it a musical? Was it a play?
It was a musical.
Oh, wow. So you sang too.
I definitely had solos. Um,
That's great. That's
I
fantastic.
was not good at it, but I,
That hence the fourth grade, and
yeah,
no updates on that.
yes,
Well,
exactly.
we are glad that that you chose science over musicals. Although I would love to see you perform in a musical if that ever comes up again. Please let us know.
Yeah, you'll be the first to be invited.
Thank you. Okay, with that, take it away, Dr. Walker.
Yeah, thank you so much for inviting me to speak. I'm really excited to be here, even with some of the technical difficulties we started out with. And I'm going to be telling you about some of the work we've been doing on how thyroid hormone might be mediating drug reward in a sex-specific manner. Oh God. Let's see. Okay. So the overarching question of my lab is, how do sex differences in substance and alcohol use disorders develop, manifest, and respond to treatment? And we've been focused on this at 3 different levels. One, how adolescent development might impact substance use disorder risk, and then both if we can find treatments for substance use and alcohol use disorder. And recently, we've really started to focus in on thyroid hormone and how thyroid hormone might be doing this. I'm going to start by telling you about some of our adolescent development work and how that is a risk factor for substance use disorder. And then later on, I'll give you some new published data that we found on alcohol use disorder vulnerability as well. So it's pretty well established that there are sex differences in the risk for substance use disorder. For men, it's generally that men tend to use drugs at a greater rate than women, and just using drugs is a risk for substance use disorder. However, when women do use, they have many other risk factors, which is they start earlier with drug use, they progress to dependence more quickly, they report greater levels of craving, and they're more likely to relapse. When they do relapse, they consume more drug during that relapse, putting them at greater risk for overdose. And they are also less likely to seek treatment. And these factors could have a lot of underlying biological and social implications. But there is data suggesting that the pharmacology of how psychostimulants impact the brain are different at the biological basis. So women have been shown to have lower amphetamine-induced striatal dopamine release. They then report less euphoric effects of amphetamines. They're more likely to have greater comorbidity with other mental health disorders. And they're also more likely to report that self-medication is a major reason for use, whereas men are more likely to report that the euphoric and risk-taking aspects of drug use are the major reason for use. But there is something that is common amongst men and women in their risk factors, and that is adolescent experience. And so why would adolescence be such a critical time for this this vulnerability to develop? Well, it's a period when the reward circuitry gets reorganized. There's a change in what we perceive as rewarding. So with regards to social reward, in childhood, the rewarding interactions are mostly through parent-child interactions. And anyone who's ever raised a teenager knows that as they move through adolescence, they move away from parents and they move towards peer-peer interactions as being the more rewarding experience. And what's important to know about this is that this is a highly conserved event, and it's critical for individuals to learn the proper behaviors to survive and reproduce as adults. We also know that the quality of these social interactions matter. So there's plenty of evidence that shows that negative or a decrease in social interactions during adolescence is predicted for reward-related neuropsychiatric disorders in adulthood. They seem to be sex-specific. There's some evidence that adolescent boys are more sensitive to this than adolescent girls. And we in the preclinical world have tried to model some of this using what's called post-weaning social isolation in rodents. And we've used it for about 30 years as a preclinical model for this neuropsychiatric risk disorder. So when I first started in this work, I wanted to ask a pretty simple question, which was, if we limit isolation specifically to the adolescent period, how does that affect behavior in both males and females in rodents? So these are mice. This is a paradigm that we use pretty consistently. The animals are shipped to the facility. They're housed for 24 hours to acclimate. And then they go into isolation from postnatally 22 to 42. So specifically during that adolescent period, they're then rehoused with their original cage mates. So the ones that they were housed with here. And then they're allowed to grow up into adults, and we check their behavior. And the first thing we did was do cocaine condition place preference. So this is just a measure of how much those animals learn to associate an environment with an injection of cocaine. And we found that male mice showed an increase in conditioned place preference for cocaine, which is something that had been shown for about 30 years. So we were pretty happy to replicate that. But what was interesting and what people hadn't shown was that there was a decrease in females. So we saw this opposite effect in males and females in their association with cocaine in the cocaine paired chamber. We then went on to look at a couple anxiety-related behaviors using elevated plus maze and marble bearing. And we saw that males spent more time in the open arm and females spent less time. Being in the open arm is an indicator of decreased anxiety because they're more exploratory, is what it's thought. Whereas males buried fewer marbles in the marble bearing task and females buried more marbles. So the way you could kind of interpret this was that social isolation, specifically during adolescence, enhanced reward and reduced anxiety in males, but reduced reward and enhanced anxiety in females. And that is one way to think about it. But because I've studied sex differences for so long, I knew that these are very sex-specific behaviors, and I tend to think about it slightly differently, which is that social isolation was resulting in a gain of sex differences in cocaine CBP. So a behavior that wasn't necessarily sex-specific becomes sex-specific, and a loss of sex differences in anxiety-related behaviors. So those animals are now moving towards the mean. And when I saw this, the first brain region I thought of was the medial amygdala as maybe the likely target of social isolation. So the medial amygdala in rodents is sexually dimorphic. It's larger in males than it is in females. It's really important in regulating these sex-specific behaviors that we see in rodents, and some of these are in humans. It is also organized by a pubertal testosterone surge, meaning that if you take away testosterone before puberty, you don't get the increase in size, even if you give testosterone back after puberty. So it's really sensitive to what's going on in the adolescent brain. Others have shown that it's sensitive to post-weaning social isolation. Bradley Cook has shown that there's a smaller medial amygdala in male rats that are isolated. And also there's reduced cellular activity in isolated males that were isolated during adolescence. And then I had shown that adolescent social isolation disrupts sex-specific transcriptional responses to cocaine. So this is one of the reasons we thought this region might be important. We then took an approach to try to determine what was going on by integrating our bioinformatic data and reveal some key sex-specific drivers of this transcriptional changes. And the one that we ended up landing on was this molecule called crystalline mu. And the reason we found this gene to be so interesting was in our bioinformatic data set, we looked for a gene that was a key driver, meaning it's central, it's connected to a bunch of other genes in the modules. But there was sex-specific connectivity. So you can see here, this is a group house male module. Crystalline mu or CRIM is right in the center. It has a lot of connections, whereas in the group house female module, it's over to the periphery. It has fewer connections. It's smaller, meaning it's not as big as a hub of those genes that it seems to be regulating. And then when we looked in our socially isolated animals, we saw sort of opposite effects. So in the socially isolated male, crystalline mu is on the periphery, it's less connected, whereas in the socially isolated female, crystalline mu is in the center, it has all these connections. And we started wondering if maybe we could target genes that are showing these kinds of patterns to impact behavior. And so the next thing we did was to just overexpress crystalline mu, which is a thyroid hormone binding protein. So it sequesters thyroid hormone in the cytoplasm of cells. We overexpressed it in adult medial amygdala to look at the behavioral effects. So this is just the paradigm we used. We didn't start overexpression until the animals were post-natal 70, but we did ship them to the facility to recreate some of the other early life experience that the animals had had. And what we found was that by blocking thyroid hormone with CRIM overexpression, we see that males show an increase in cocaine-related behavior similar to the isolated animals. They spend more time in the open arms of an elevated plasmid, similar to the isolated animals. And while we didn't see the effect in females on reward, we did see the anxiety-related phenotype. We then went on to sequence these to see what transcripts were changed, particularly because thyroid hormone and its receptor are transcription factors. Oh, I forgot to mention, we overexpressed it in the nucleus accumbens and saw no effect as well. So this suggests that the medial amygdala is really the brain region that's regulating it. And we just looked at the transcripts that were regulated by crystalline mu in the males on top and the females on bottom. Blue means those genes are downregulated. Yellow means the genes are upregulated. And these are all of the genes that are regulated by CRIM. And you can see that the profiles in males and females are very different. There's more in the male than there are in the females. And in fact, the directionality is very different. So we wondered at the transcriptome-wide level what might be happening. And to answer this question, we used a technique called rank-rank hypergeometric overlap that just tells us what direction all the genes in the transcriptome are going. So you can think of this as if the genes are going down in the same comparisons, you're going to see heat in this window. If the genes are going up in both comparisons, you're going to see heat in this window. And if they're going in opposite directions, you would see heat in this direction. And what we found that was really interesting was by overexpressing crystalline mu in the male medial amygdala, we saw very consistent overlap with genes that are regulated by cocaine, suggesting that in the male medial amygdala, but less so in the female medial amygdala, blocking thyroid hormone may have a similar transcriptional profile to just giving an injection of cocaine. And this, the way we interpreted this was that the medial amygdala is a critical regulator of sex differences of anxiety and reward, and that thyroid hormone signaling within the medial amygdala may be modulating these sex-specific behaviors through transcriptional regulation. So why are we so focused on transcriptional regulation? Well, like I said, thyroid hormone is a transcription factor. The way it works in the cell is thyroid hormone goes in, binds its receptor, the receptor homodimerizes and activates transcription at thyroid hormone response elements or genes that are responsive to thyroid hormone. So in our CRIM model, what we think is happening is that when you overexpress CRIM, it sucks up the thyroid hormone, keeps it from going into the nucleus, and then thyroid hormone receptor signaling goes into what's called ligand-independent signaling, where there's heterodimers, it recruits a bunch of repressive proteins, and it turns off genes that are thyroid hormone responsive. So to test this, we went back to our sequencing data to say, well, if this is really happening, is what we should see is that transcriptional repression is occurring when we overexpress crystalline mu. And in fact, that is what we saw in males, that there were 87% of the transcripts that were regulated by crystalline mu overexpression were downregulated, as shown here in blue, but only 13% were upregulated. Whereas in the females, we saw that there was kind of a 50-50 split in the number of genes that were up and down regulated. And just as a reminder, the males were the ones that showed an increase in cocaine CPP after CRIM overexpression. So this made us start to think that maybe some of these transcriptional profiles might be important in mediating these behaviors. So the next question we had was, if this is really true, then we should see that thyroid hormone-mediated gene expression is influenced by social isolation. So we went back to our original data set, and we just looked to see how thyroid hormone responsive genes might be changed in males after adolescent social isolation. So this is sort of an overview of how thyroid hormone works in the brain, just because it's a little more complicated. It involves conversion of the active form into astrocytes, and then that gets transported in neurons and drives transcription. And when we looked at how thyroid hormone-mediated genes were responding, we saw very few differences at baseline. So socially isolated males and control males had pretty similar transcriptional profiles of thyroid hormone-responsive genes. But when we layered on cocaine on top of it is where we really started to see the differences. So the socially isolated males are mediating a lot more transcriptional targets that in response to cocaine than those control males. And importantly, we saw no effects in the females. So this gave us some indication that thyroid hormone responsive genes are involved in these behaviors that are mediated by social isolation. But what is actually really interesting is there's very few studies that have looked to see if cocaine even influences thyroid hormone and if thyroid hormone even influences cocaine. So when I first started my lab, we wanted to take a step back and try to better understand what might be going on. And we asked if there's a relationship between thyroid hormone signaling and cocaine reward. And so this is just a reminder of the entire thyroid hormone axis here, where TRH is released from the hypothalamus. It acts at the pituitary gland to drive thyroid stimulating hormone, or TSH, which then acts at the thyroid gland to drive the release of T4 or T3. T4 is the inactive form. And then DIO2, deiodinase 2, acts on T4 to convert into T3, and that's where you get the active forms in the nucleus. There's also plenty of evidence in the clinical literature that suggests that thyroid hormone dysregulation is associated with many neuropsychiatric disorders, including anxiety and depression, alcohol use and substance use disorders, bipolar disorder, and PTSD. But the preclinical data was really lacking. And so we started with probably the most simple experiment you could think of, which was to take our animals, give them either 10 doses of saline or 9 doses of saline and then 1 dose of cocaine, euthanize them 1 hour later, and see if their thyroid hormones are changed in the serum. And we looked at gene expression to see across the different brain regions, are there changes in thyroid hormone responsive genes? We included a no injection control, and this will be evident when you first see the serum data. So first, this work was done by Teresa Vu and 2 summer students, Alina and Amelia. When we looked at thyroid stimulating hormone in response to cocaine, we saw a big sex difference in that males have higher TSH when they're injected with saline, but lower TSH when they're injected with cocaine, whereas females have really low TSH in response to both of these. This does get carried over into serum T4, but only in males, where there's a reduction in serum T4 in both saline and cocaine injected animals. And then T3 is down in the animals after injection regardless when compared to their control animals. So you can see that in females, the negative feedback system seems to be working. But in males, particularly when we look at the saline injected versus cocaine injection, we're starting to lose what we would predict the negative feedback mechanisms would look like. So we then wondered if any of these would be sort of reflected in gene expression changes. And to do this, we took that tissue and used qPCR to measure gene expression in the prefrontal cortex, the nucleus accumbens, the basolateral amygdala, and the medial amygdala. We chose these based on their role in reward and cue-associated effects on substance use disorder. And the first thing we wanted to look at was just CFOS to see, make sure that we were actually getting gene expression that we predicted. So did we actually change activity-dependent gene expression? And interestingly, in the males, so the males over here that I just highlighted in blue, we do see across these brain regions that there are specific changes in CFOS, but the females are not changing their levels of expression of CFOS across many of these brain regions, which we thought was pretty surprising, given that we had already seen some other changes in thyroid hormones. We then went on to ask if genes that are associated with thyroid hormone metabolism might be affected across these brain regions. And again, just as a reminder, DIO2 is the enzyme that converts T4 to T3, and then DIO3 is the enzyme that breaks down T3 into its inactive form. And then crystalline mu is the one that binds it and keeps it from interacting in the nucleus. So for the prefrontal cortex, we saw sex and sex-specific effects where there was an increase in DIO2 in the males and females, but only a slight increase in females for DIO3. We also saw this sex difference in DIO3 that hasn't been reported before. So we interpret this to mean that if DIO2 is going up, that's converting T4 into T3 and increasing the levels of T3 in the prefrontal cortex of both males and females. When we looked at the nucleus accumbens, on the other hand, we really didn't see a whole lot of effects, which was pretty surprising. But DIO3 goes up with the injection. So we interpret that to mean, since this is the gene that breaks down thyroid hormone, we interpret that as a reduction in T3 in the male and female nucleus accumbens. The BLA seems to have really sex-specific effects where DIO2 goes up with cocaine in males, but crystalline mu goes down in females. And what's interesting here is that these two mechanisms are going to lead to the same changes in T3 in the BLA, but through very different methods, right? This is increasing the amount of active, and this would be a decrease in sequestration of T3. And then finally, the medial amygdala, we saw all three of these enzymes sort of changing with cocaine. DIO2 is up. DIO3 shows a similar pattern to our TSH changes, and then crystalline mu goes up here, which we interpret to mean that while you're getting more T3, you're also sequestering more T3, and maybe there's no change in thyroid hormone within the medial amygdala that you would see in other brain regions. We looked at the receptor and we saw no effects in the BLA or the nucleus accumbens, and we saw a few pretty minor, minimal effects in prefrontal cortex, thyroid hormone receptor alpha. There's a sex difference, and this THR goes down with saline and cocaine, whereas no effects are observed in THR beta. And then in the medial amygdala, we see an increase in both the saline and cocaine injected animals for THR beta. So we saw these changes in thyroid hormone responsive genes sort of in a very region-specific way and a sex-specific way. And we wanted to know if we manipulate thyroid hormone, does that then impact cocaine reward? And so to do this, we took adults, we gave them Synthroid or synthetic T4 in their water for 15 days before we did behavior. And then we just measured their preference for cocaine. And what was interesting was that even though we saw these sex-specific effects, when we just increased the levels of circulating T4, we increased the preference of cocaine in adult males and females, regardless of, or across the sexes. We also saw that there was no effect of antagonizing thyroid hormone by treating them with PTU, suggesting that this is a specific cocaine-induced or a change in cocaine reward that doesn't happen when we block thyroid hormone. So to summarize what I've shown you so far, social isolation dysregulates the maturation of the thyroid hormone responsive genes and they're responsive to cocaine. There seems to be a bidirectional regulation of thyroid hormone and cocaine sensitivity in adults where cocaine changes thyroid hormone and changing thyroid hormone changes cocaine sensitivity. Acute cocaine exposure disrupts thyroid hormone-related genes in a sex and region-specific manner. This is particularly true for the medial amygdala and prefrontal cortex. And pharmacological dysregulation of thyroid hormone increases the preference for cocaine across the sexes. And then I think this is probably the biggest takeaway from this is that if we can use models where sex differences are disrupted, either in behavior or transcription, we can leverage that disruption to better understand and identify novel biological pathways that are imparting this risk for substance use disorder. And so that's really what we've relied on is using these models where we know sex differences in behavior are disrupted to better understand and identify precision treatments for substance use disorder. So I'm going to switch directions now to talk about our work on alcohol use disorder because this work has been really, really exciting, and it's something that I never thought I would be doing, was alcohol-related work. But the data is so compelling that it's the direction that we're taking the lab now. So when I first started, there was a student who came into the lab. He was very interested in alcohol use disorder, and he was very interested in thyroid hormone. And we know that there's some pretty strong clinical evidence that suggests that chronic alcohol dysregulates thyroid hormone signaling in humans. Most commonly, it seems to reduce T3 and T4 and can have effects on the thyroid hormone itself. There's some rodent models that also suggest that thyroid hormone is disrupted by chronic alcohol exposure. But what we hadn't seen was if animals were voluntarily consuming alcohol, how does that impact the thyroid hormone axis? So this is work done by Michael Johnson, who's a PhD student in the lab, and in collaboration with Dr. Andre Binion, who's an alcohol use disorder researcher here at OHSU and an expert in these behavioral models. And we started out by just using a very simple two-bottle choice where the animal can choose between ethanol and water. They had 30 total days of drinking where the ethanol concentration is stepped up. And then we collected serum to measure thyroid hormone levels. And then we looked at the hypothalamus for thyroid hormone responsive genes. This was recently published, so I'm not going to go into a ton of it, but I will give you the take home. This is just showing that they do, in fact, drink. Females are the dotted lines. Solid lines are the males. Females drink more ethanol than males. We looked at serum T3, or we looked at serum thyroid hormone levels and saw a few changes, and saw a few changes in gene expression, except that we saw that ethanol increases serum T3 and decreases THR beta expression. And this made us wonder if potentially alcohol might be disrupting negative feedback through THR beta. THR beta is the receptor that's pretty well known for modulating negative feedback in the hypothalamus. And so we wondered if we can target THR beta, can we then change alcohol drinking? And we did this experiment in collaboration with Dr. Tom Scanlon here at OHSU, who's a thyroid hormone receptor expert. And he's developed a drug called SOBAM2 that is specific to the brain. So you inject SOBAM2 into the body. Sobeterome is the active form of this drug. It doesn't get activated until it gets in the brain. So this is just showing there's way more SOBAM2 in the brain than there is in the serum. And then we can specifically target THR beta in the CNS without having the peripheral effects of thyroid hormone agonism. The other important thing to take from this is that SOBAM2 is in phase 2 clinical trials for depression and bipolar disorder. So we already have some evidence that this is pretty safe in humans, which gives us we're really excited that maybe we could take this in for alcohol-related work too. And Michael did a very simple experiment where he got the animals drinking, and then he started treating them with SOBAM2, and then we looked to see how their drinking changed. So once again, the dotted lines are the females, the solid lines are the males, purple are the vehicle-treated animals, orange are the SOBAM2-treated animals. We started out with a lower dose of SOBAM2 on intake. And while we started to see some effects in males, we really didn't see anything in females until we bumped the dose up to 1 mg per kg. So you can follow these orange lines and you see once you get to 1 mg per kg, they precipitously drop off in ethanol intake. This is shown here in our summary slide where baseline drinking versus day 6 of treatment versus the last day of treatment, you see a decrease in their intake in ethanol in both males and females. And we also see the ethanol preference goes down as well. So they're not, so they're showing less preference for alcohol while they're not drinking as much. So this was pretty exciting to us. And we then wanted to know, though, is the reason these females are not changing their drinking until we get to the higher dose because we just needed a longer time? There's some evidence that these drugs take a while to have their effect because they're transcription factors and they need to get into these sort of steady state systems. So Michael did another really big experiment where instead of changing the dose, we just took it out for much longer. And we euthanized half the animals at day 9 of treatment, and we euthanized the other half at day 23. And you can see here that by day 9, you start to see a decrease, particularly in the males, in their ethanol intake. And it stays low throughout the rest of the experiment. Whereas in the females, it sort of bounces around a little bit, suggesting that this low dose of SOBAM2, while it might decrease ethanol intake, it's more effective in males than it is in females. And again, this is really highlighted in our summary data where you see that from baseline to treatment day 9, there's a really precipitous drop in the drinking in males, where that's not observed in the females, and it stays low out to 23 days in males, but not females. We did see some effect on preference in the females, though, so that does tell us that maybe there is an effect. It just might not be as potent in the females than the males. And so we're really trying to understand what might be happening with these different doses of SOBAM2 and how we can better target drinking across the sexes. So because thyroid hormone is a metabolic signal, there's a lot of ways that SOBAM2 could be reducing drinking, and we wanted to address some of those before we got too far down the pathway. So one, it could be altering metabolism. There's some evidence that it affects feeding, so we wanted to make sure it wasn't reducing feeding. It could be that it was decreasing reward, particularly rewarding properties of alcohol. There's also evidence in humans and rodents that messing with thyroid hormone can impact taste. So we wanted to make sure this wasn't, they just like didn't like the taste of alcohol. So to address this, the first thing we looked at was body weight. There are very few changes in body weight over time. There might be a tiny minimal increase in the middle of this experiment, but nothing that we got that really tipped us off that this was a change in metabolism. We also looked at ethanol clearance at 2 different doses, both 2 grams and 3 grams per kg. And we found that the animals are clearing ethanol at the same rate, whether they're treated with SOBAM2 or with vehicle. And then Michael took on this experiment where he looked at food intake, he looked at sucrose preference, and he looked at quinine taste avoidance to determine how these other factors might be impacted by SOBAM2 treatment. I'm going to go through this kind of quickly because most of it's negative data. So first of all, we didn't see any change in food intake across this entire experiment. So this is before they're treated, and then middle of treatment and the end of treatment, there was no effect. When we looked at taste aversion, we did this at 2 different doses, and I'm just showing you the high dose of quinine. And one of the reasons I want to show you this is we do see, this is pretty consistent, that SOBAM2 treatment increases water intake. So we don't see any change in quinine intake. We do see a slight increase in water intake, and this does show that there's no change in quinine preference. So they're not avoiding quinine at a significantly different rate. We saw similar effects at a low quinine concentration of 0.03%. And then when we looked at sucrose preference, we did this at 2 different doses as well. We saw that, once again, there was a slight uptake in their water intake, but when we looked at preference, there was a decrease in sucrose preference. So they were not drinking they weren't choosing sucrose over water in both males and females. And we saw similar effects at a high sucrose concentration of 2%. We also saw this effect when we use palatable food. I'm not going to go into all of these. So we do see that when we give them a choice of sucrose or a sugar pellet, they eat less sucrose when they're choosing between either chow or when they're choosing between water and sucrose. So one of the ways we've been thinking about this is that it could be reducing the rewarding properties of ethanol, although we need to do a bit more investigation to make sure, especially because we start to see this increase in water intake in all of our studies. So now the question is, if we give them SOBAM2, is this changing gene expression in these reward-related brain regions that could explain how we're seeing these differences? And then can we leverage this to better understand why males and females aren't responding to that lower dose of SOBAM2 in the same way? So to do this, we took those animals that were given the high dose treatment of SOBAM2, and we just did bulk RNA sequencing of the prefrontal cortex and the medial amygdala. And the first thing we found was that in the prefrontal cortex, these data are all pretty preliminary, but they're really exciting, and so that's why I put them in here. The first thing we found was in the prefrontal cortex, we saw no changes in gene expression when we compared ethanol to water, regardless of if the animals were treated with SOBAM2 or vehicle, and regardless of if they were males or females, which was pretty surprising that ethanol was not changing gene expression. But when we looked at the SOBAM2 treated animals, we saw huge changes in gene expression whether or not the animals were drinking ethanol or whether they were drinking water. We did see that there were more genes that were regulated by SOBAM2 in the males than in the females, and this is particularly true for those ethanol drinking females. So we did a real quick comparison. Oh, sorry. We did a really quick comparison of the Go terms that were associated with some of these genes. I'm not going to go into all of these because we'd be here all day and it would probably be pretty boring. But just to give you some idea of what genes might be targeted by treating with SOBAM2, we see astrocyte changes. The one that really got us excited was this glutamatergic synapse. There's some evidence that thyroid hormone can influence synaptic plasticity. So seeing some of these changes is really nice. Also, oligodendrocyte precursors. We've seen some changes in myelination with thyroid hormone. So all of these are sort of in line with what we would expect, and they're sort of confirming that that these are targeting the Go terms that we were predicting. When we decided to use RHO to compare how transcription across the entire prefrontal cortex is impacted, this was one of the things that really surprised us. So we first started with those males that were either treated with SOBAM2 and drinking ethanol or treated with SOBAM2 and drinking water. And you can see that SOBAM2 is having really similar transcriptional changes across these two comparisons. So it's SOBAM2 that's driving the transcriptional changes, regardless of whether or not they're drinking ethanol or water. But in the females, there's much less overlap of those transcriptional profiles. So this once again points to how different the transcriptional profiles are across the sexes and across brain regions. And I think it's something we really need to spend more time thinking about, and how these transcriptional patterns can be so different across the sexes, and to better understand how we could target and treat some of these sex-specific disorders. And then finally, again, this is pretty preliminary, but this once again made me really excited when we looked at it. These are the genes that are regulated by SOBAM2 in the prefrontal cortex. I just want to point out that there's kind of an equal number of genes that are upregulated and downregulated, with the exception of these females treated with drinking ethanol. But when we looked at the medial amygdala, which is being run by another grad student in the lab, Kelly Abshire, the genes, there's a lot more genes that are upregulated by SOBAM2 across these groups than downregulated. This is true for males drinking ethanol and water and females drinking ethanol and water. And when we just calculated like the percent upregulation, you can see that in the prefrontal cortex, the average is about 59% of those genes are upregulated, whereas in the medial amygdala, 72% of these are upregulated. This is associated with a reduced ethanol preference. And if you remember from earlier in my talk, when we overexpress crystalline mu, which is blocking thyroid hormone, we caused more transcriptional repression and enhanced cocaine preference. So this is a really nice comparison, and this is one of the hypotheses I've had for a really long time, is that if we increase transcriptional repression in the medial amygdala, we can change drug reward. And this is showing that if we upregulate it, we can also change drug reward, which is giving us something to think about in how transcription and how upregulation and downregulation of genes can lead to behavioral changes. So just to summarize what we've seen here, thyroid hormone signaling is disrupted by alcohol in rodents. Manipulating thyroid hormone with this CNS-specific thyromimetic reduces ethanol consumption. There are sex and dose-specific effects of SOBAM2, though, that we really need to better understand. It's seemingly acting by reducing the rewarding properties of ethanol, although this might be complicated by the fact that they're drinking more water, so we need a better understanding of what's going on there. And there are robust transcriptional changes in the prefrontal cortex and the medial amygdala, which are induced by SOBAM2. So what we're currently working on now is to determine the transcriptional targets of SOBAM2 in both the medial amygdala and prefrontal cortex, and determine if SOBAM2 can have similar impacts on cocaine preference. And we're also treating the SOBAM2 during adolescence to see if we can block the persistent behavioral and transcriptional effects of adolescent social isolation. And so, just to take a step back, I want to sort of talk about why we think this is happening. Um, Going back to these behaviors that we know can be sex specific, particularly in rodents. These behaviors that are sex specific are, one, necessary for the perpetuation of many species. They often emerge during adolescence. They are very energetically costly, and those energy costs differ across the sexes. And so, what do I mean by that? Like, let's take copulation as an example here. So, for both males and females, copulation and a copulatory bout is an incredibly energetically costly event. But for males, once the copulatory bout is over, the energy demands are pretty much gone. However, in females, assuming that that copulatory bout ends with a pregnancy, those energy demands are just beginning. And so we really do think that thyroid hormone, like a lot of these findings that we're seeing where it's really sex specific and females are less sensitive to some of these thyroid hormone changes, are probably driven by the differences in energy demands of these sex specific motivated behaviors. And to better understand that would help us better target different systems in treating substance use disorder vulnerability. And with that, I'd like to acknowledge all the past members of the lab, as well as the current members of the lab. Kelly did the medial amygdala sequencing. Mason actually just graduated. I haven't had the heart to move him over to the past members quite yet, because he just graduated in September. Mandy and Teresa are both research technicians. Teresa is looking to go to grad school in the upcoming year, so look out for her application. And Michael did all the SOBAM2 alcohol behavioral work, and he is also on the job market. So if you're looking for a postdoc and you do alcohol work, you should keep him in mind. And then finally, I'd like to thank our collaborators, Dr. Andre Ryabinian and Dr. Tom Scanlon, who were instrumental in getting these alcohol-related work off the ground. I did just get a new R01 from NIDA starting in September. So we'll be able to continue a lot of this work. And we also have funding from the Circle of Giving Women's Health Grant here at OHSU, as well as the Medical Research Foundation of Oregon. And with that, I will happily take questions. I'm assuming I should stop sharing my screen, or?
It's totally up to you.
Okay.
You get to do what you want. You're the guest.
Nice. I'll stop
Thank you so much. Thank you everyone for giving your little emojis, clapping emojis. That's the hardest part about these virtual things, is that you don't get the instant reward, but it's nice to see those little floating clappy things. And congratulations on the R01. That's really great, especially in this climate of
Yeah, it was getting a little dicey there, but
that's great though. Good news for a lot of reasons, which is
yeah.
fantastic. So I have a couple questions. Please either put your hand up or feel free to unmute yourself and ask a question. Otherwise, you're going to hear me asking questions, which is not as much fun, but I'll do it. And people are sometimes nervous, but I will start, and then you guys can put something in the chat or just unmute yourself. So one of the questions I had, first of all, it was really, really fantastic, and I really enjoyed it. And I tweeted, I'm sorry, I'm not supposed to say tweeting anymore. I posted on Blue Sky none of the stuff that was in prep, but I tried to get it as accurately as possible, but I'm sure I made a few little mistakes. A very potentially boring question at the beginning, which is which is because you mentioned at the beginning, of course, that while men tend to be more likely to be users of substance use, women report like less euphoria with amphetamine. And then I'm thinking about the work of Jill Becker showing that it can change that the dopamine release can change across the estrous cycle and possibly menstrual cycle in women. So I'm wondering, did you look at either estrous cycle or I told you it
Yeah.
was potentially boring.
No, that's actually somebody asked the same
Oh,
question,
of course.
so I can answer it twice. Um, okay, so the short answer is yes, we've looked at estrous cycle effects across a lot of these endpoints.
Yeah.
The longer, more complicated answer is um, for the adolescent social isolation stuff, we've never seen estrous cycle as a big component of it, but for all of the sequencing data, we euthanized those animals when they were in diestrus
Right. So you
to
know,
try to
yeah,
hold cycle
yeah,
steady.
yeah,
And the reason we did that, and I do think this is like kind of even like the sex differences we saw were still pretty fascinating. The reason
yeah.
we did that was we were thinking that the motivational drive in a female during estrus and proestrus is quite different
Hmm.
based on the fact that she's like,
Of course.
leaving her
Yeah.
nest and going out and finding a copulatory
Partners.
partner.
Yeah.
And so we were trying to sort of bring those a little bit more in line to better understand what the cocaine-induced transcriptional profile would look like.
Sure.
We still saw really big
Yeah.
differences
Of
across the
course. Yeah. Yeah.
domains.
Yeah.
So even like holding that steady and thinking we were like keeping them more in line, we still saw really big differences. That was
Yeah.
kind of surprising.
Yeah.
The crim effects, I have tried to figure out everything that could explain why those females look different, and it's definitely not associated with their cycle. It could be, I mean, we've done factor analysis. I've done like correlation analysis. I've done everything I can think of to try to figure out why they're so different. And I just haven't been able to figure it out.
You
And
mean
then,
in terms of their dose response? Because I didn't even on the high dose, like you can see a split right away with the
yeah,
males. You don't see that. Like they
yeah,
they kind of dance around a bit and then they start coming down.
yeah. So I don't know
Yeah.
why it doesn't seem to be linked to
It's
their
not to the cycle.
cycle.
Yeah. Yeah.
Yeah, it
Yeah.
doesn't seem to be cycle, but I don't exactly know why it doesn't change. Um, the SOBAM2, we don't smear them throughout the entire experiment, but we do take their smears at the like when we euthanize them.
Hmm.
So we have them staged, and we haven't seen any
Any
effect
lineage
of.
effects.
They're still cycling. We still see
Yeah.
all of the different
Hmm.
representations. So
Hmm.
it doesn't seem that SOBAM2 is just like completely obliterating their cycle.
Hmm.
But I don't know if there's some underlying like subtleties that we haven't been able to pick up.
So in the Sobium 2, I was trying to remember how to say it.
Yeah.
I'm kind of mimicked what you're saying. Experiments, you didn't have social isolation stress on top of that, right? Are these in adults then? Are
Yeah, these are
they
just adults
just
that we
plain, yeah,
were. Yeah, so I mean, I must admit, like I was a little skeptical when Michael came into the lab and was like, I want to do thyroid hormone and alcohol because I've never done alcohol
right.
work.
Yeah, yeah,
Did like work out that it was kind of like a perfect, collaborative effort with
yeah,
Andre and Michael sort of pushing that together.
yeah,
Um, I was hopeful, but I was like, I don't know this. And then the fact that we had Tom here who had developed this like
yeah,
CNS specific
yeah.
agonist,
It's pretty amazing, actually.
kismet, right? It's like,
Yeah,
I can't believe this like came together and worked out.
yeah. Yeah, no, it's great. It's a really nice story. And
Yeah.
this, it just strikes me that it's um, which I think has plagued science, not plagued, it's good. That, it's a beautiful story, like full circle in the males, but the females are really much more difficult to decipher in terms of what. So do you have any other, so, but it's possible that's just not thyroid hormone mediated. There are some
Yeah.
signs that it is, but do you have any other ideas of what might be contributing and to, because
I
females,
think,
yeah, go ahead, sorry.
oh, sorry. I think, and Michael thinks this is true, is that because estrogen can compete with
Yeah,
thyroid hormone,
right,
you can have estrogen receptors
right,
out competing
right,
thyroid hormone
right,
receptors at the DNA.
yeah.
Um,
So would you try to do something like Laura isn't exactly suggesting this, but like would you OVEX them and see if
Yeah, that's definitely one possibility. The other thing we've found though is like it seemed, especially with the like low versus high dose SOBAM2,
Hmm.
it seems that if you really hit the thyroid
Yeah,
hormone system
then you can get
harder,
it. Yeah,
then
yeah,
you can get it,
yeah,
right? And so it could just be a dose response thing where
yeah.
you
But it's
have to get thyroid hormone like
really high. Yeah.
high enough to outcompete the estrogen receptors.
Hmm.
But it's something we're like really working on. And in fact, Kelly has the nice thing that we can answer with Kelly's experiment that I didn't go into because we haven't quite gotten all the data analyzed is she's got high dose males and females. So they show the decrease in drinking, right?
Hmm.
Got low dose males and females where the males show the decrease in drinking, but the
But
females
not
don't.
females.
And then
Yeah,
she's just got ethanol drinkers.
right.
And the goal of that study is to use, you know, how I was saying, like, use these disruptions in sex specific behaviors to better understand basic
Hmm.
biological
Hmm.
mechanisms.
Hmm.
So use those females that aren't responding to low
Right.
dose and see where the transcriptional profiles are different
Hmm.
in the high versus low dose females and see where they're the same in the high versus low dose males.
Hmm.
Because that should
That
help
could be
us,
cool. That could
yeah,
be
that
really
should help
cool,
us
right?
extract
Hmm.
what those targets are and
Hmm.
what is really like potentially
Hmm.
changing the drinking behavior.
Yeah, so you must have some, I mean, you don't have a lot of animals, but you must have some, oh, my microphone switched. I guess it doesn't matter because you guys don't notice. Um, uh, you must have some animals that drink more and some animals that drink less. So you could sort of do like a responder, non-responder
Yeah.
kind of analysis
Yeah. I
too.
mean, the males that are treated with sodium too, just like,
Like just they drop. Yeah.
yeah, they just
I
drop.
noticed that. They, like there was a really nice separation in the
Yeah.
males.
Yeah.
And the
Um,
females, I mean, I only remember the one, but I remember that sort of like for the first 2 days, they were like kind of
yeah.
flip-flopping a bit, and but then they drop, but still not to the same kind of.
Yeah. They never go like all the way down to where the males are. And so I think that's something we just have to like try to parse in our molecular data. Um, and potentially, I mean, I don't know if we would ever, I don't know if you could give a high enough dose of SOBAM2 to get them to like come down to normal levels.
Right. And what, and yeah, well,
Starting to have
some
really,
other,
yeah, like really weird off-target effects, right?
yeah.
So
So, um,
I think we'd
uh,
be better off finding other targets than just
right. That's what I'm wondering. Like, is there something
yeah.
else, which I think those other experiments will give you perhaps some clues anyway. I think
Yeah.
is really, really interesting. And I also just want to commend you for a lot of people would stop at a certain, I know not you, but other people would stop like using, females because it's, well, we see the phenotype in males, and so we're just going to keep going. But I think it's really, really important to keep them in there. And you still see, like I was at one point I was like, oh, you're gonna you're gonna show that it's working only in males, but there were some instances where you did see some effects in the females too. So I was
Yeah.
like, I thought that was really, really important. Um, and just going back to, I can't read my own writing here, but one question I can read is, do you see, and sorry, this is at the beginning of your talk, but do you see changes in social isolation? Does it have any changes in terms of puberty onset? Or
That's
do you look at that?
yeah, I did look at that. It does so one nice thing about C57s is they go through puberty, like there's not a huge sex difference in the timing
Yeah.
of puberty.
Oh, I didn't know that. Hmm.
So unlike rats where there's like
Yes.
15-day
Yes. Yeah.
difference, uh C57s are like 28 days in females and 30 days in males.
Oh,
It's like
so it's not
so
that. Yeah,
yeah.
yeah, yeah. It's pretty
It
tight.
does at least we've shown this
Accelerate it,
like
maybe.
a few times where it advances the
Yeah, expel. Yeah,
onset of puberty.
yeah.
We stopped tracking it because
It's such a long term
well,
thing. Yeah,
it's really subjective. And
yeah,
so what we were
yeah,
getting, like when I did it all by myself
yeah.
was fine. But if anyone came in to like track puberty that wasn't me, or like we couldn't have the same person doing it all the time, the
Right.
data started to get real, especially
Wonky.
males.
Yeah, yeah.
Yeah.
Oh, interesting.
Also kind of in females, like female mice, they their vaginal opening isn't like as
As dramatic.
yeah, as dramatic as it is in rats.
Hmm. Hmm.
And so there would be like one
Interesting.
person would be like, yeah, she was open
Ah.
yesterday. We got a smear. And then the next day, like, she's closed. And it was,
It's like, no, this is not possible. What's going
yeah,
on? Yeah.
yeah, it became like a little I started getting nervous that like it was just too
Yeah, and
subjective.
you're doing it's a pretty, it's a long time period, so it's not
Yeah.
likely going to affect much. Okay, so now we are out of time. So I
Yeah.
want to just thank you again. It was
Sure.
really, really fantastic, and you stretched my brain in lots of different ways. Thank you for doing that on December 8th, after I spent, thinking about sugar plums all weekend. So it was very good for my brain. Jolt
Yeah.
me back. And let's just give her a last little emoji round. If you can, yes, thank you so much. I'm very
Yeah.
happy that you can do that. And thank you so much for agreeing to do this.
Of course it
And
was.
congrats on your RO1.
Yeah.
And
Yeah.
let us know about the musical.
I will. I will. I will. All right. Well, thank you for having me. This was really fun. So,
Everyone, my name is I guess I'll use the Dr. Lisa Galea, and I'm the lead of the Women's Health Research Cluster. And I'm really delighted today to introduce you all to one of my favorite people, Dr. Deena Walker. And I know she's going to give us a fantastic talk on thyroid hormones. And I don't know very much about thyroid hormones, so I'm excited about that. But before we listen to her, I want to recognize that we live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Toronto, which is covered by the Toronto Purchase Number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And I know not all of you are on Turtle Island, so I encourage all of you to learn about the Indigenous peoples on lands of which you dwell. A little bit about the Women's Health Research Cluster. We have actually over 1,000 members now across 43 countries, which is pretty amazing, and we are here to promote and catalyze women's health research across all disciplines. We run a number of events, a trainee research presentation series, so if you're a trainee and interested in presenting, please look out for calls for that. We run a number of travel awards. We just had an award lottery. It's a lottery system, unfortunately, now, but hey, you never know. So they run twice a year. So please be on the lookout for that. Become a member and support us if you're not already a member. And with that, I'm going to stop sharing my screen so that I can read out Dr. Walker's bio. How's your computer doing? Is it in the middle of your presentation?
it's back. It's fine.
Yay! Okay. Um, so while you start sharing, I will read your bio and your fun fact.
Okay.
I made I made Deena change her fun fact for you. Deena Walker is an assistant professor in the Department of Behavioral Neuroscience at the Oregon Health and Science University. She's passionate about understanding how hormones influence brain and behavior, so am I, and is dedicated to using her diverse training background to identify precision-based treatments for neuropsychiatric disorders, particularly for women. Currently, her lab is focused on how hormones influence sex-specific vulnerability to alcohol use disorder and substance use disorder. She moved to Portland, Oregon in 2020, an auspicious year, after doing her postdoctoral fellowship at the Icahn School of Medicine at Mount Sinai, where she studied the molecular mechanisms of sex differences in addiction. As a PhD student at the University of Texas in Austin, she studied how the brain controls ovulation and menopause with a specific focus on hormonal regulation of gene expression. Such exciting work. In recognition of her innovative and impactful science, she has been awarded several early career awards, including the Frank A. Beach Early Career Investigator Award from the Society for Behavioral Neuroendocrinology in 2023. In addition to her laboratory work, Dr. Walker is active in science outreach and storytelling. Her stories about the impact of her own life experiences on scientific questions that have been featured on the podcast StoryCollider. Now, her fun fact is that she was the lead